Hi everyone, welcome to the Carbon Copies Foundation Journal Club meeting for March 2020. My name is Anita Fowler and I am an aspiring biomedical student devoted to the goal of substrate independence and whole brain emulation. I will try to be thorough but keep this presentation short and sweet so ample time can be spent on discussion. So the paper I'll be discussing is an argument by Robert McIntyre on the website of his organization NECDOME called The Case for Glutaraldehyde, Structural Encoding and Preservation of Long-Term Memory. Now, I chose this paper because whatever the stuff of ourselves is made of, it most certainly includes, if not exists within, the umbrella of memory. This paper is based on current memory research discussing existing theories of memory, what is involved in them, and whether or not they may be preserved with existing technology. So to give you a little background, Robert McIntyre, back in 2015, along with Gregory Fay, published a paper introducing aldehyde-stabilized cryopreservation as a means to preserve the brain's ultrastructure. This led him to joining 21st Century Medicine and winning the Brain Preservation Foundation Prize for successfully preserving a full pig connectome using this technique and using glutaraldehyde. Since then, McIntyre has seemed eager to keep the ball rolling and move to human brains by starting his organization, NECDOME. Though there has been some pushback toward the idea of being able to store the brain's memories for future emulation, McIntyre seems determined to do the research necessary to show whether or not this method of preservation will actually store memories, whether what we think of as a fully preserved connectome is enough, basically if the information is there. And that brings us to McIntyre's hypothesis. That is that glutaraldehyde fixation may preserve the information that encodes an organism's long-term memories. And when he says memory, he is referring to all of the following: semantic, long-term, and episodic memory. To be more specific, McIntyre defines long-term memory as, quote, observable changes in behavior that persist for longer than 24 hours, including semantic memories, long-term motor skills, and episodic memories. I'm not exactly sure why he chose 24 hours versus 2 or 12, like in some of the studies he references, but 24 is what he has decided on for his definition. The first point that McIntyre discusses is that memory is physically robust, withstanding traumatic events like temporal global ischemia, where the brain loses its extracellular space, and deep hypothermic circulatory arrest, where all electrical activity is lost. In both these cases, the long-term memories of humans and other mammals have been shown to return after successful recovery. Additionally, within the component parts currently thought to make up memory, there can be some loss. Research with engrams shows that they can survive even when some of the neurons or synapses are lost, either due to natural decay or external destruction. In his paper, McIntyre reviews evidence of memory encoding as durable changes in synaptic structure, protein distribution, gene expression, and other physical changes, all of which are discussed in some detail through the concept of cycles and engrams. McIntyre shows that for long-term memory formation to occur, it means that um, distribution of neural proteins, especially synaptic proteins, must change, and that the changes must occur at multiple neurons and synapses. As a result, we see the occurrence of self-reinforcing cycles and engram formation. Now, to go into a little more detail about what he means about a self-reinforcing cycle, He cites what he refers to as the AMPA cycle, involving receptors and molecules at glutamatergic synapses, all of which he listed in his paper. So before I continue though, I'd like to briefly explain part of this cycle for those who are unfamiliar to drive this concept home. This is a vastly oversimplified before and after drawing of a synapse during long-term potentiation, or when signals are such that we will form long-term memories. So we start out on the left with a synapse with a sort of spindly looking dendrite spine. And what you see in yellow is the neurotransmitter glutamate being released from one neuron, triggering the opening of an AMPA receptor on the other neuron. Now, an AMPA receptor is a protein in the cell membrane that allows positively charged sodium ions to come into the cell. If enough sodium gets through to make the inside of the cell positively charged enough, then the magnesium ion in blue will be released, opening an NMDA receptor, which is shown in pink. When that receptor is open, calcium ions are then able to enter the cell. Why these things flow the way they do and everything involves further details about ion channels and gradients, and we won't get into that now. But suffice it to say that when the calcium gets in, some exciting things happen with the protein kinases and transcription that basically lead to making those AMPA receptors more sensitive, making more of them. So they allow more sodium to come in so that more calcium can come in so that they can make more receptors and on and on and on. So you end up with stronger synapses like the one on the right with a more mushroom-like dendrite spine and more going on. So we have a cycle that perpetuates and strengthens itself. And what's great is that even if a molecule or some small part is lost, the cell can still continue, or the cycle can still continue. These cycles perpetuate also in the soma of the cell to influence the making of protein products, further signaling, and even epigenetic modifications. But put another way, McIntyre references the research of Chen et al., saying that information is encoded, quote, redundantly in the states of mutually reinforcing macromolecules such as proteins, mRNAs, nanostructural components, and even modifications to DNA. Now, what's extra exciting and interesting about this reference is that although the most commonly held and most referenced in McIntyre's paper memory models are based in synaptic theories of memory, these particular researchers represent a genomic theory of memory. So I feel like McIntyre is definitely covering all of his bases here. So to summarize, the summarizing factor here is that despite individual molecule loss, these are self-reinforcing cycles, and, quote, the cycle itself is able to maintain its state almost indefinitely. Now, let's zoom out from the neuron to neuron relationship, and we'll see what happens when a whole assembly of neurons alter their synaptic structure during memory encoding. What we get are called engrams. Now, if you're not already familiar with engrams, when they occur, they create or are a pattern that can easily be accessed or recreated, used, and updated at a later time. To give a better understanding, I grabbed this figure from the Jocelyn et al. 2015 paper, Finding the Engram, which McIntyre cites. On the left, you see the encoding of an engram where there is a strengthening of connections between collections of neurons activated during the event. Those are shown in red. This is then consolidated over time so that it can more easily be recreated, allowing the memory to be retrieved. Now, when the engram is reactivated, there is a time of destabilization during which new information can be added and the engram can be adapted. It is then restabilized and through reconsolidation can go dormant and later be retrieved yet again. So, like the self-reinforcing cycles discussed earlier, engrams too can survive the destruction of some of their parts. Some of the research McIntyre cites includes studies on tagging and manipulation of engrams, erasing long-term memories, and even creating false memories in mice. So, when we ask ourselves, how do memories survive? Whether it's day to day life or traumatic events like global ischemia or DHCA, we can know based on this research that, quote, biochemical cycles and the physical redundancy of engrams play a big role. Okay, so bringing all that back to how we can preserve all of this. How can we successfully stop in time these processes without losing the vital data that would one day perhaps enable us to use our earned knowledge of memory and integrate it with future technology that might be able to retrieve it? Um, McIntyre argues that glutaraldehyde is the candidate that may help us preserve the information that makes up memory. The pros he discusses are these. Um, it reacts rapidly to form crosslinks with tissue, creating a stable gel-like form resistant to decay. It can withstand major changes in pH, temperature, osmotic stress, etc. He pulls on research with other aldehydes as well, especially formaldehyde, to show the durability of aldehyde stabilized tissue. Proteins and mRNAs can be identified, labeled, and analyzed after the fact. Glutaraldehyde is primarily used currently to prep tissue for electron microscopy because of its preservation ability. Um, all biomolecules in the AMPA cycle are verifiably retained. And if you're curious, he actually links to a study for each and every molecule in his paper. Also, McIntyre notes that with a level of detail, the glutaraldehyde creates, it, quote, creates an injective mapping between durable memory states and preserved artifacts, end quote, which, quote, preserves comprehensive information about long-term memories. So, with all this good news, how could anything be wrong? McIntyre does link to some drawbacks of glutaraldehyde. There is a loss of extracellular space. However, this may not actually be a problem, as memories are shown to be recoverable in such cases, like with cerebral ischemia. And because of the dense crosslinks, visualization with probes is encumbered. However, with ever improving slice and scan techniques, I wonder if this may be less of an issue if we're talking about destructive scan and copy for whole brain emulation. So to summarize all of this, I'll share some of McIntyre's arguments. One, it appears that all potential components within current memory theories, both synaptic and genomic, are able to be preserved with glutaraldehyde. Two, the noise induced by glutaraldehyde fixation appears to be less than what is used to create long-term memories. And three, that there are no current viable theories of memory that would account for the robustness of long-term memories that would not also be preserved with glutaraldehyde fixation. Now, with all that said, I know that there is intense interest by many in this method succeeding, and I'd be interested in knowing what may have been missed or overlooked. Additionally, I believe this was posted in late 2018 or early 2019, and there has been since further study in memory. With that said, I would like to open up the space to discussion, other theories, ideas, counter arguments, updates on the research, etc.
Thank you. That was really fantastic. What a great presentation of the material.
Thank you.
At the risk of jumping the queue, I was going to just I was going to say two things. First of all, I had the pleasure of sitting down yesterday with Robert, actually, it so happened, and we discussed a bunch of things about Nictome. And one of the things he mentioned that he would still like to achieve is he would like to make glutaraldehyde fixation stable at a higher temperature, because right now Um, the samples still need to be preserved at, I think, something like minus 138 degrees Celsius, and that is more expensive than he would like. It's not a cheap way to preserve samples. So there's that. But other than that, it seems like everything is going very, very well right now in terms of technical hurdles or challenges to overcome. The other thing is that I just kind of wanted to ask you if you knew what the main complaints were, or the main challenges from others that he was trying to overcome with that paper. Do you know why this paper was written? In what context?
I don't feel like I can say definitively because I haven't talked to him. However, there were a number of articles that were critical, either because there were theories of memory that involved like broader electrical cycles that seem largely dismissed in the scientific literature, but nonetheless did create a little bit of pushback. And additionally, just the idea that Necto may be claiming, hey, we can preserve human brains and bring them back at a later date and everything's going to be great. I think the press got a hold of some of that and maybe took it a little bit further than was desirable. And so, since McIntyre has been like, hey, we're doing research here, we're trying to figure this out. We know we have a good thing here, but we got to make sure if with current theories of memories that everything that we're claiming can be preserved, and it can be used for uh, different forms of research, not just the idea of hopeful whole brain emulation. And certainly that he's not guaranteeing that we will be able to emulate a brain. So I think he was basically trying to curb some of that and say, we are a research organization. This is some of the culmination of our research of what memory is and how glutaraldehyde may be able to preserve those memory traces.
All right. Yeah. So, I know that there are some other theories of memory out there, although they aren't very strongly supported. It seems to me like what Nectome is doing should be enough to preserve memory. It feels almost like the only other major argument out there is kind of the one that we'll be discussing at the workshop that's coming up, which is this whole question about personal identity and which metaphysics you buy into. As long as the memory can be preserved, that's really the only other thing to worry about besides reconstruction, that whole technical issue. How do you create an emulation out of that? Yeah.
Yeah, I mean, even the most recent paper that I've looked at, it was just from late last year. It was about synaptic clustering and memory formation, and it was more of a consolidation of theories, but still, I believe, would be preserved with glutaraldehyde. So, or at least with the long-term goal of what it'll be able to do.
Mm-hmm. Is there anyone else with questions? I don't want to dominate the floor.
I did have a question. How long after a patient is deceased? How long how long do they have before the all of the components start to break down before they're unsavable? What's the maximum amount of time a deceased patient can go before they're being given the glutaraldehyde treatment before they actually start losing those components that define their memories?
So from what I've read, there can start, I mean, it depends on the cause of death, I suppose, but there can start to be loss really, really quickly after. And so, there are a lot of techniques with cooling of the body and everything to make it last a little bit longer. But I know that some of what I've read, um, that Hayworth has posted from Brain Preservation Foundation, talks about almost trying to administer it as the part of the dying process, like freezing the brain before any decay could happen. If anybody understood that differently, please let me know. But that was my understanding of what I read.
Yeah,
Yeah,
that's right.
and I was going to say, in the original studies that Robert published, when he did the pig brain, the pig was actually alive, and I mean, obviously under anesthesia, and that is when they injected the glutaraldehyde. It's kind of started the process while the pig was still alive. So, that's the way the protocol is designed, but I don't think no one really knows, you know, because the problem in humans is, if you go back to the history of cryonics, you have to wait till they're legally dead and then try to rush in and do procedures. And that, that's a huge open question. So the ideal way to do it and what Robert's working on is people actually doing, assisted dying and, going right into, as they're still living, the heart is still beating, go right into, the glutaraldehyde procedure.
Right.
Thank you. I see. So this is in a scenario when this would be used to, like for instance, when this is in a scenario when this is readily available, it would be more applicable for those who are on their deathbed and they're being given the treatment rather than just like say a patient dies in a car accident and then brought to the facility. That's probably a lot of time already to lose memories. Okay, I see.
Exactly.
Thank you.
Yeah. Yeah, I know that Hayworth published a protocol where like a person is diagnosed with Alzheimer's and they kind of choose a point in the future before the decay becomes too much that they will begin the process right then to salvage as much as possible.
Yeah, and that's what Robert is trying to do. Is actually get at some point, get a case of someone who's dying. And in the Netherlands, actually, they have done assisted dying in people with dementia. You just have to obviously do it early enough where the person can, give consent. But they do that. I don't say all the time. I mean, there's been 100 something cases. That's still kind of a niche thing. I mean, that's still there's a lot of questions about how um, besides that, like you said, people die in an accident. How long can you preserve um, tissue? And I mean, I think there's a bunch of open questions, and I think that hopefully there's some leeway that you don't have to do this instantly. But
And there's evidence as well, if you're in a cooler environment where the temperature is already lower and that's where you pass away, the degradation may be a little bit slower, so there may be a little bit more time. Whereas a place that's a normal warm temperature, it could happen within minutes.
Okay. Um, and I do have another question then. Um, how long can glutaraldehyde preserve uh, a brain in its like classifiably original state? Like, is it like decades or centuries? I'm curious. Like, what's the time scale there?
I think the consensus is on centuries.
Oh,
I don't know that we know when it will stop.
yeah, if you lower it to the with the
Right.
cryonics, if you just keep it at room temperature, maybe a few months. I mean, no one knows for sure.
But it's not super sensitive also. So for example, once you fix a piece of tissue and you've had it down to some temperature, you can take it out and you can keep it in a refrigerator for a few days, or you can even keep it outside on a shelf for a few hours, and it's not going to do anything perceptible to the sample whatsoever, at least according to what Robert told me yesterday.
Yeah, because he's thawed samples out for a couple days and then put them back in the, the cryonic temperatures, and then um, back and forth a few times. So you've definitely got some room. But yeah, long-term storage, it has to be whatever it was, minus whatever 100 degrees or whatever.
So I think what we're really missing at the moment is we're missing any further peer review and sort of that feedback loop that usually gets started whenever there's been a new advance. We're not the experts, obviously, but somebody who is an expert in the same technologies, in the same technique, in the same neurobiology, I expect that at some point there would be a response to the articles that Robert has produced, and that there would be another cycle of at least investigating whatever the critiques are, if there are some, or just taking it to the next level. I haven't really seen that yet.
Well, I think that's that's what Robert is trying to do, and also Ken is just, you've got to get the neuroscience community to take you, seriously first before they're going to argue with you, and I think that's what they're working on. That's the biggest challenge, probably, is because people are always saying, "Oh, we don't know how the brain works, and we don't know," and of course, Ken or Robert really pushes back on people when they say that, and kind of backs them into corner pretty quickly, but. I think that's the key, is just get the neuroscience community to start debating, kind of debating these things, which hopefully we're getting there. I mean, it does seem like, slowly, some of these ideas are taking off, but I know it's been a struggle to get, neuroscientists to, and that's what Robert would say. If somebody says you don't think it works, well, tell me why. Let's what specifically do you see as an issue? But he also needs to publish. I mean, he really needs to put some of these things out there in bigger journals, and and I think that would help too.
I do really like the angle that he's taking on how he's pushing this now, because instead of focusing on the question of reconstruction and on whether or not people can survive this way, he's focusing more on just the preservation of memory, and preservation of knowledge. And it's very hard to argue that you wouldn't be preserving, in some sense, the knowledge that is in a person's brain by preserving the material of the brain. In all likelihood, there's some way to get at that information in the future. It's sort of like how you would, maybe back when we first realized what DNA was all about, if we had started preserving DNA at that time, just to build a bank of DNA that might otherwise be gone.
Yeah, I agree. I think that's a good strategy, because you pin it to us I think that's what Ken is doing with the aspirational prize too, is just focusing on memory first, and then once you convince people of that, I think then you've got a much better shot at the whole why bother to preserve your brain kind of questions.
If I recall correctly, I do think that McIntyre is starting to receive like some like cadaver brains, that are able to be used in research through Nectome, so that at least pieces can be preserved that way, potentially used for research later, if I'm not mistaken.
I don't want to misquote him, so I don't want to say anything that I'm not absolutely sure of, but I think you're right that he certainly has access to some samples. That's not the same as saying that anyone has actually been preserved with this method for the purpose of storage. I wouldn't want to go that far. Although it seems like he is gearing up to offering the service, or offering at least a subscription to the service.
Well, I agree. I would love to see some more publications come out of his research and his group.
So Anita, I take it that you personally have no problem with the copy problem. Let's just say you don't think there's any issue in storing someone's brain and recreating them from that, or personally your views on that?
No. At this point, I would round my concerns for the idea of successful scan and copy to zero. And there are things that are interesting about like the idea of the self. How do we identify the self? How can I be reassured that on this new substrate I would still have the experience of being me? And yeah, that's a kind of a tough one. Like how can we prove that before we try?
Yeah.
But I feel fairly confident that it seems like a worthwhile direction to go. I don't have a lot of qualms with that.
Okay. Yeah, this was just sort of a side question of mine because the workshop is coming up next weekend, and there's that question. But more to the point of Nectome and Robert and all of that, I understand that what Nectome is hoping to do is to do full body of preservation using this procedure rather than, say, neuro only or brains only, the way that has often happened at Alcor. And I asked Robert why he would do that. And that's kind of where the story veers away a little bit from the story about just preserving knowledge or preserving memory. Because the reason why he would want to preserve whole body is because he feels that even if you are doing whole brain emulation and uploading after this, if you scan in the entire body and you create an emulation of the body as well as the brain, then you won't have to go through physiotherapy once you're there again. So his concern is that if you take just the brain and then you create an artificial new body for a person, that then they will have to go through essentially physiotherapy to become used to using the body. Now, I didn't debate that any further with him. We didn't talk about whether there's any accelerated ways to do that once you have access to all of the nerves and everything. So we didn't really get into that in any depth, but it seems like that was the reason why he's thinking of doing whole body, despite the fact that preserving the glutaraldehyde stabilized tissue at that temperature that they have to stabilize right now is the major cost, is a big cost burden. And of course, if it's a whole body, then that's a larger volume that you have to store. You can't store as many in the same volume as you would if it was neuro only. So that was an interesting little detail I found.
Yeah, I think that's very interesting. I mean, that is one of the big concerns is like how do we, maybe we have all of this stuff stored in our brain, but how do we cue it back into life if we don't have the normal inputs that we've been used to having? And from like a systems perspective, our body is made up of a lot of systems and a lot of neurons, not just in our brain, and they're connected. And without the ones that we're used to, getting the ball rolling, so to speak, for recognition might be a little bit more difficult. So I could see the appeal of having the whole body preserved as well.
I'm a little. I have a question about like preserving the whole body for physiotherapy. I'm not clearly understanding like in what form that would mean. Like if you, like, for example, if you're emulated as just a digital self, I don't see how like physiotherapy after emulation would be necessary. So I mean, is he like is he referring to physiotherapy after emulation in some way like that, or what is?
Okay, so I'll be honest that when Robert mentioned this, at first I was very confused, and I thought that maybe he meant that we had to be brought back in biology, that you had to reverse the glutaraldehyde fixation process and somehow revive people the way that many at Alcor want to be revived. But that was not his point. His point was that the precise way in which the brain is communicating with the muscles in your body through all of the peripheral nervous system and the rest of your your nerves and connections in the spine and elsewhere in the body. That is complex, and if you've had an accident or you haven't used your muscles for a while, or you had a transplant, let's say you've received someone else's finger or something, then you may need to go through physiotherapy to get used to using that limb or using those limbs. And if it's an entire body, then I guess that could be quite a bit. It might almost be like you've been paralyzed for a week or something like that, or a month, and now suddenly you need to learn how to do everything again. So that's where we didn't get into any deep discussion, because while I think he's right that if you were to naively approach this as, okay, we're just going to reconstruct your brain, and then we're going to take an artificial body and we're going to connect that directly, and then the brain's just going to have to figure out how to work with it, and the brain has to do all the learning, which is what we would do today, right? If we just plug an artificial limb into, say, the motor area of the brain or something, or into the cerebellum or something like that. If we wanted to do something like that today, then indeed that would involve physiotherapy. I have a hunch, but it's no more than a hunch, that that's going to be a little different when we're talking about a system where you have access, because you've done an upload, an emulation of the brain, where you have access to every single new artificial neuron, whether it's simulated in a computer or built in some other fashion, and you could more intelligently engineer what I would call the translator between that and whatever your body is going to be. So it may not involve the same sort of physiotherapy. Sure, there might be, this 5-minute thing for each of your limbs where you need to figure out how the controller works, and you figure that out very quickly because the controller adapts to your output signals. Your brain is producing output signals, and that gets transformed into what you're supposed to do with your body. So I can imagine that there would be engineered solutions that can be more efficient than physiotherapy. But those are just hunches, and I've never really looked into it. So I get his point, at least, yeah. But yeah, on the flip side, it means then you have to do an emulation of the body as well. And that's not super easy either. You have to reconstruct all that too.
Thank you. Okay. I did have one final question that I had, as far as which relates to identity, but then also like where the boundary of identity is, because we're talking about preserving the memory. My question is preserving the memory is more risky than the rest of the brain? Because as I understand it, you could pinpoint the memory in the hippocampus, but it seems like for identity, if you're concerned about like what makes up one's mind, then there's also like where the consciousness resides, which if I got this right, it's in the frontal cortex. Does the entire brain does that just preserve the same, or could you say that the hippocampus is more vulnerable to like degradation after death, especially if like the memories are the most vulnerable thing? Is that a factor?
Somebody else would probably do better to add to this, as I can only answer so much of that. But I think that preserving the whole brain in part is important, because some of what you said I'm not sure has been proven, such as where consciousness actually sits in the brain, and there's also multiple areas of the brain where memories are stored, depending on which point of what level of creation they're at, and even that, it seems, based on my research, to be an open topic of research. So preserving the memories alone, I'm not sure if we could even do that without preserving a lot of other stuff at this point.
Yeah, I also think this runs into the problem of what people call memory. The definition of memory is, it's a little vague when people don't say what they mean. So, in a layperson's vocabulary. When we say memory, then we say something like, oh yeah, I remember that party yesterday, or oh, I remember what a car looks like. And then we say that's a memory. But memory is also how you move your fingers when you play piano, the particular way that you walk, your gait, the way you pronounce things, the way that you process what you're seeing when you see a red car, and how that associates with memories that you have of red and of cars and of that time when you went on a holiday somewhere, and then what that evokes, so that whole downstream propagation that happens. So the reality is that that it's more than just what we normally think of as memory when you're playing a memory game. It is all of the places where there has been a change in the brain because of an experience that you went through, because of your life and your living. Not just that, of course, the blueprint as well, what you started with when you were born, but then add to that all of those changes and every synaptic change that's happened. All of that, in a sense, is memory. And so when you talk to a physicist and you ask them, what is memory? They'll say, well, it's any process where if you look at the function, it's got hysteresis in it. So in other words, once an activity has happened, the next time the activity happens, the function will produce a slightly different output because of the activity that happened before. Then there's memory in that function. There's memory in that system. That's memory very much at its foundation. And so, yeah, you can't just carve out a piece of the brain and say, we only need this bit, unless we've discovered that there are parts that are so general that you can just put in a general purpose piece of that brain, and the rest of the brain will work with it quite easily. And in fact, that's actually something where the hippocampus may be a good candidate, because the hippocampus has cells that are not required perpetually for the memories that they are learning. After a certain period of time, your memory, as it becomes long-term memory, becomes independent of reactivation in the hippocampus, and those same cells are reused to learn new things. So you could almost say that's a general purpose resource, a pool of cells that can temporarily store pointers to patterns of activity elsewhere in the brain. So you'll have some of those pieces in the brain where perhaps at some point we can decide that you might as well make them general purpose and not care so much about reconstructing them specifically. But then again, if we're reconstructing 90% of the brain specifically, then maybe the rest of the 10% isn't a big deal either. So then you just do the whole thing. Could go either way, I think.
Yeah, I think to kind of go off of that, like I feel like I'm of two minds. On the one hand, it would be nice to preserve as little as possible and only have the stuff that's absolutely necessary, and whatever redundancy exists across humans can just be recreated in general in an emulation. But just like with the issue with the body, it may be easier for the brain to adjust to an emulated environment if it has the component parts, including maybe some of these pieces in the brain that may be multi-purpose. I don't know.
Yeah, there are so many strange things we can explore once we have some emulations to work with, even if they are just emulations of very small animals like C. elegans or Drosophila. Because another thing that will be really interesting to future developers on whole brain emulation is, what if we can make the whole system a bit more clean? Let's say instead of using neurons that fire unreliably or synapses that fire unreliably and this probabilistic soup that's in there, what if we had units that actually really preserve memory and you never really forget memory? What if we can clean it all up and in the end you basically just need a tenth of the cells that are there if these are a certain type of reliable unit and it works just a little differently? If the experience is still the same, if it turns out after experimentation, after study, we find out that this doesn't change, the sense of being, the sense of being yourself, it could be that there are a lot of ways of translating, simplifying, optimizing pieces of how the brain works as part of a process of enhancing who we are and how we work. And that's all super speculative, but it's the sort of thing that I imagine is possible when you really have access to the entire system. And you can learn from it, and you can study it, and you can play with it. You can try out things, and when something isn't as you desire, you can back out. You can, you've tried that little thing, you decide that you don't want it. It's like if you try, I don't know, a medication that turns out to have side effects you didn't like, then you don't take the medication anymore. Something along those lines.
You make it sound like a really cool, like, human mind video game.
Well, that's what I think is going to happen. If we can choose our own self-directed evolution because we have access to everything and we can directly work with it, then I think what you get is this, what I often call next Cambrian explosion of possible directions, branches in which people can choose to develop. I think there will be a huge ecosystem of new intelligences in a sense. But yeah, it's so hard to sort of even predict anything there. I think maybe this is room for science fiction writers to explore a little bit, to give us some idea of what might be possible.
Actually, Keith Wiley might argue with me a little bit on this one, but I love the idea of with the whole branching personalities to solve kind of the problem of first truth. Like if you read a book first and then you watch a movie, it colors your experience of the movie, but then you never get the experience of like watching a movie first, which would then color the experience of the book. But if you can, have two different ones that go out into the world and experience it differently, then remerge later, then you solve that problem and can have one truth, but from two different pasts.
Yeah, I have wondered a lot about merging people, how that could be accomplished. It's an interesting thought, merging back together. I'm not actually sure if Keith was even is Keith here for this journal club?
Bad day, maybe not.
Oh well. So I don't know, Michael, do you think that this is the time where we thank Anita for her wonderful contribution, or are there still people waiting to ask questions? Do you see anything come in?
Let's see, let me check. We don't have any further questions. So yeah, I guess with that, unless anyone has any other questions at the moment, thank you very much, Anita. It was a pleasure, and it was a great discussion.
Thank you, guys.
And with that, I will go ahead and end the stream right now.